Okay, so we're going to continue looking at the solution properties of polymers. Now, when polymers are when polymers are introduced in a solution, they exhibit a given viscosity. Now, viscosity is designated by this symbol here, like N with a tail. Now, the viscosity of these polymers that are in solution, they depend on a number of factors. So, they're dependent on the concentration of the polymer, the polymer solvent interactions. So this is how does your polymer behave in a given solvent. And then the third one, also the charge interactions. So this one mostly affects polymers that are charged or polymers that are ionized. So as the ionization of these polymers increases, or as the ionization decreases, so it also has an effect on viscosity. And then apart from that, the binding of small molecules to your polymer in solution can also affect the viscosity. Now, what are we saying when we say what is viscosity? So we're saying viscosity simply is the resistance of a flow of a solution, the resistance to flow of a solution. Now, viscosity is divided into three, or it can be described in three ways. So there's what we call relative viscosity, specific viscosity, and also intrinsic viscosity. Now, what is relative viscosity? So relative viscosity, which is given by, which is designated by this shortcut. So you have the symbol for viscosity and REL, just for a shortcut for relative. So this is relative viscosity. It is simply just the ratio of the viscosity of the solution to that of the pure solvent. So which is here, this is the viscosity of the solution to that of the pure solvent. So what are we saying? So you have a pure solvent where you're going to introduce your polymer, which is designated by some green substance here. So first you have a solvent. So this solvent where you're going to put your polymer can have its own viscosity. So this is the viscosity of the pure solvent. And then when you put your polymer in this solvent, you form a solution, because sometimes these polymers can dissolve. So you form a solution. Now this solution which is formed is going to have a different viscosity to that of a pure solvent. So that's what we're saying. So relative viscosity is simply just the ratio of the viscosity of the solution to that of the pure solvent. And then what is specific viscosity? Okay, so specific viscosity, it's simply just relative viscosity subtracted by 1. Okay, and then the other one, which is of most importance, is the intrinsic viscosity. So relative viscosity, you can also call it just as an absolute viscosity. So the other one is the intrinsic viscosity. So intrinsic viscosity is just a measure of how much your solute is contributing to the viscosity of your solution. So that's what intrinsic viscosity is. So it is designated by viscosity, the symbol for viscosity, but it should be in brackets. So I'm saying it's just the measure of the solute's contribution to the viscosity of the solution. And then we're saying that intrinsic viscosity, how is it found? How is intrinsic viscosity found? So intrinsic viscosity, it is found by plotting a graph. So as you can see, so it is found by plotting a graph of the ratio of specific viscosity divided by the concentration of your polymer against the polymer. Okay, so these so this is what is called the reduced viscosity. So you have the specific viscosity to the concentration of the polymer. When this is plotted against the concentration, you're going to get let's say a graph. Now extrapolation of that graph should be extrapolated to concentration 0, and the value that you find on the y-axis is what is called the intrinsic viscosity. Okay, now sometimes when a polymer is introduced in a solvent, they tend to form spherical particles. And if it's not all the time, but when and if your polymer is introduced in a solvent and it forms spherical particles, then the relative viscosity is calculated as 1 plus 2.5 multiplied by volume fraction. 1 plus 2.5 multiplied by volume fraction. So what is this volume fraction? So volume fraction is the volume of the particles that are going to be in this solution divided by the total volume of the solution. Okay, so that is how you calculate the relative viscosity for polymers which, when they dissolve in solution, they form spherical particles. Now, such that, how do we define this equation? Such that now a plot of the specific viscosity to the ratio, a plot of the ratio of specific viscosity to the volume fraction against the volume fraction. So it's just like what we're doing here. Against the volume fraction, you're going to get a plot. And then when you extend this plot, or when you extrapolate, so when you extrapolate it up to here, this value you find here, it's supposed to be a 2.5. So it's supposed to match what is in the equation. If your polymer in solution contains spherical particles, so this value, which is the y-intercept, should be 2.5. Now, if there's any deviation from this value, as long as a polymer is forming spherical particles in solution, if there's any deviation from the 2.5 after you plot your graph, there are two explanations to it. It's either your particles are hydrated, your polymer spherical particles are becoming hydrated. So it's due to particle hydration or particle asymmetry. Particle asymmetry simply means they are not, your particles, they don't have the, an equal shape and also like in size. So there's particle asymmetry and also particle hydration. Sometimes it can be both the factors. So both these factors can result in deviation from this value. Now, another thing that we looked at, we said that polymers can change depending on which solvent they are in. What was that? So we said one of the factors that affect viscosity is the polymer solvent interactions. Now, we're saying that there can be a change in the shape of the polymer depending on the type of the solvent polymer interactions that occur. So when you introduce your polymer in a solution, your polymer can change its shape depending on the interactions that are occurring between your polymer and the solvent. So sometimes it can just be stretched out, and sometimes your polymer is going to be forced to coil up. Okay, and then we're also saying that and because of this like change in shape of your polymer, so depending on how it's going to interact with your solvent, the shape might change, and the change in this shape results it affects the viscosity significantly. So let's see how that happens. So I'm saying there's a good solvent, and then there's a bad solvent. So there's a good solvent, and then there's a bad solvent. Now, in a good solvent, we're saying that your polymer chain, it's for linear macromolecules, they tend to expand. Okay, they tend to stretch. Why? Because they have functional groups that that match with the solvent, or that have an affinity for the solvent. So we're saying in a good solvent, we call something a good solvent when when your polymer and the solvent have a stronger force of interaction, or when they can interact. So it means your polymer has certain functional groups, okay, that can easily interact with this water. So they have a greater affinity for each other, such that your polymer, it expands in this solvent and it's stretched out. So as it expands and is stretched out, the viscosity increases. However, when your polymer is introduced in a bad solvent, so what you're calling a bad solvent is that your polymer and the solvent, they do not have a greater affinity for each other. Now, because of this, so what will happen is that, so for example, if this is a non-polar solvent, but then your polymer is, let's say it has polar groups and non-polar groups, so it's going to coil in such a manner that the inside part of it is going to be the polar, okay, and then the outer parts, these chains are going to be non-polar. You see, like poles to like poles. So the polar part is going to be shielded from the non-polar environment, and then the non-polar parts are the ones that are going to be exposed to this environment. So the whole point is, in a good solvent where your polymer has good affinity with your solvent, they expand, they are freely, they are flexible, and so viscosity increases because there's greater interaction here. But here, there's no greater interaction between, or there's literally no interaction between your polymer and the solvent. And so it's like they're still separated, but just in one place. So the solvent here, the viscosity here reduces. As you can see here, it increases. And then the other one that we looked at, we said sometimes the charge on these polymers, or the ionization of these polymers, can also affect viscosity. Now, how does this happen? So we're saying that viscosity of ionized polymers, it is also affected, or it's dependent on the charge interactions of these polymers. So we're saying polymers that are charged, they may also vary in shape depending on the ionization, okay? Such that at maximum ionization, so if your polymer is charged, and mostly this one occurs for the linear macromolecular polymers. So if your polymer is charged in nature, okay? So that means if your polymer is ionized, so at maximum ionization, or as the charge of your polymer increases in solution, that means also as the concentration of your polymer increases in solution, so they tend to be stretched out. Why do they stretch out? Because of the mutual charge repulsion. Okay, because there's increase in the same charge, so this tends to be this repulsion. So they tend to be stretched out in solution due to the mutual charge repulsion, and as we established, so it means this is going to increase the viscosity. Now, if by any chance we add, let's say, some counter ions, so counter ions, these are substances that are going to be cancelling out the charges or the maximum ionization. So addition of any counter ions, it means it's going to reduce the charge. So let's say if the maximum ionization was positive, we're going to start adding negatively charged substances, such that they start to cancel each other out. Now, addition of such counter ions results in reduction in the charge. If there's reduction in the charge, it means the ionization is reduced. Reduction in the ionization, it means that your polymer chains are going to contract instead of them being stretched out. And if they contract, it means viscosity is going to reduce. Now, we're saying that the intrinsic viscosity of most of the linear macromolecular polymers is directly related to their molecular weight. So the greater the molecular weight of these large polymers, the greater the intrinsic viscosity. Now, this intrinsic viscosity for these linear macromolecular polymers, or these polymers that have high molecular weight and are linear in shape, it is determined using the Stondiger's equation, which says intrinsic viscosity of these polymers is given by K, which is just the constant of the solution, or the constant of the polymer solvent system, multiplied by M, which is of course the molecular weight, then to the power A. A is just a constant for these high molecular weight polymers, which mostly will range from 0 to 2. But most of these molecular weight polymers are going to have this constant between 0.6 to 0.8. Now, one other thing to talk about is when a polymer is in solution, they have an ability to form gels, and hence bringing us to the next one, which is gels. When polymers are introduced in solution, they have an ability to form gels. Okay, they have an ability to form gels, like the normal gels that you see, the diclofenac gel, the gels for hair, and all that. Now, what is a gel? What is a gel? I'm sure we've all seen a gel. What is a gel? Now, a gel can be defined as a polymer solvent system containing a three-dimensional network which can be formed by swelling of solid polymers. So it means you can form a gel is simply just a system which contains your polymer and a solvent network. And how is this network? This network has a three-dimensional structure. How are they formed? Because they are formed by your polymer. When it's introduced in a right solvent, they begin to swell as they absorb or as they take in the solvent. And then sometimes these gels can be formed by reduction in the solubility of the polymer in solution. So I'm saying sometimes the way to form a gel is just by introducing your polymer in a solution, and then it swells. Okay, so as it swells, it begins to increase in consistency and hence forms a gel. And then sometimes the way we can form these gels is by reducing the solubility of a polymer in a solution. You can form a gel. Now, one other thing to note is that when gels are formed from solutions, each system is characterized by a critical concentration of gelation point. Now this gelation. So what we're saying here is that as you begin to form your gel, so as you introduce your polymer in a solution, so you're going to reach a concentration where the solution begins to gel. Now that concentration of the polymer at which the solution begins to gel is called the gel point. Okay, such that below this concentration a gel cannot be formed. So an exact concentration at which your polymer begins to form a gel in solution is called the gel point. At which below this point, or at which below this concentration, a gel cannot be formed. And then we're saying gelation is characterized by a large increase in viscosity. So you're going to note, how are you going to notice that a gel is being formed? So your solution begins to increase in a viscosity, which is the resistance to flow. So the flow becomes less, the consistency, it becomes thick. So at the gel point, and also as the concentration increases, we're saying the solution is going to be characterized by a large increase in viscosity above the gel point, and then your solution is going to have the appearance of a rubber-like elasticity, just the way the gel is. Then you know that your gel is being formed. And then now we're saying gels can be divided into two. So you can have irreversible gels or reversible systems. How do we determine which one is reversible and which one is irreversible? So this is just this is done depending on the nature of the bonds between the polymer chains in solution. Okay, so let's look at some of the properties of these gels. So we're saying gels can either be type 1 gels or type 2 gels. Okay, type 1 or type 2 gels. Now, how, like I said, how do we know which one are type 1 and type 2 gels? This classification is just based on the nature of the bonds between the polymer chains in solution. So for type 1 gels, these are irreversible gels. So these are reversible gels and they have a three-dimensional structure or shape, okay, which is conferred by the presence of the cross-linking which is in the system. So we're saying these are irreversible three-dimensional structures, they are characterized by covalent bonds. So the polymer chains in solution, you know, as they begin to gel, they are held by strong covalent bonds. So hence that's why they're irreversible. And then another thing to note is that when polymerization occurs, or when monomers, you know, begin to polymerize, they are enhanced by cross-linking agents. So if you remember when you in the previous lecture there was on the types of polymers, sometimes they can be linear, they can be branched, or they can be cross-linked polymers. So sometimes, so under type 1 gels, when polymerization is occurring, it is done in the presence of cross-linking agents, such that when these polymers are introduced in water, okay, they don't dissolve, but they just swell in water. Why? Because these cross-linking agents make them stable. Now, and because of their ability to just expand in water, these type of polymers, you know, they have been made, you know, as expanding implants, not as gels, but just the ability of a polymer, you know, to expand in the presence of water. So they have been made or have been formulated as expanding implants that have an ability to absorb water in the body and then swell to a predetermined size. Sometimes they can just be used for delivery of the drug. And then the other one is the type 2 gels. So likewise, these are reversible gels. So they are reversible on heating. So when these gels are heat, they can be reversible compared to these. And these are held by the weaker intermolecular forces, which are the hydrogen bonds. And that's why even reversibility is easier compared to here where you have the hydrogen bonds. And then we're saying that the temperature at which the gelation occurs, or the temperature at which the gel begins to form, like I said, is called the gel point. Now, when it comes to the type 2 gels, they can be formulated either by cooling the solution. Okay, so you're going to put your polymer in a solvent to form a solution. So sometimes as you cool this solution, okay, you can form a gel. For example, polyvinyl alcohol. So this is the PVA. For example, polyvinyl alcohol, when it's this is a polymer. So when it's introduced in water and then it's cooled at a specific temperature, a gel is formed. And then sometimes type 2 gels can be formulated by heating. So when, for example, the use of this water-soluble polymer, methyl cellulose, which is also written as MC. So when methyl cellulose is introduced in water, and then you heat the solution to a specific temperature, it can induce the formation of a gel. So that's it on this. And then we're saying that, you know, apart from us doing the heating and the cooling to form these gels, we're saying that gelation can also be influenced or can also result by addition of certain additives, okay, to the polymer solution. So addition of additives like borax. When borax is added to polyvinyl alcohol, it facilitates the formation of a gel, because it acts as a bridging molecule. And also addition of certain solvents like glycerol. So glycerol has a very high viscosity. So when it's mixed, let's say, with solvents like polyvinyl alcohol, they form a gel. Now, it's because of this consistency of gels that polymers like polyvinyl alcohol have been used in the formulation of a medicated gel. Why? Because when they are applied to the skin, you know, within just a few minutes, they dry. It dries up, leaving a thin film which traps the drug, you know, close to the skin and keeping it there for a longer period of time. Now, the other thing to look at is syneresis. Now, syneresis is one of the factors that is associated with gels. Syneresis is, you can call it like, it's also like a whipping effect. So this is where, so this is the separation of the solvent phase from the gel. How did we define a gel? So I say this is a network of a polymer and a solvent. okay, and then they form this thick three-dimensional consistency. Now, sometimes the solvent can separate from the gel, and when this happens, the process of separation of the solvent system from the gel is called syneresis, or the whipping effect. So how does this happen, just in a nutshell? So we're saying that, how do we make a gel? As we're forming a gel, it means your polymer is taking in water so that it can swell. Then as it swells, the viscosity increases. Now at equilibrium, okay, so the contracting force of your polymer, because as so you're adding water to your polymer and your polymer begins to swell. Why? Because it's taking in water, such that at equilibrium, so it's forced to expand. Now you know that as something is forced to expand, it also has a natural force to contract. Okay, so what happens is that, so at equilibrium, the contracting force of your polymer is balanced by the swelling force, which is also dependent on the osmotic pressure, or like the water being contained in your polymer. Okay, such that if the osmotic pressure is tempered, okay, it means now there's going to be an imbalance between the swelling force of your polymer and the contracting force of your polymer, such that now the water is going to be squeezed out. So when the osmotic pressure is decreased, for example, by cooling of the system, or by changing the ionization of the polymer which is in solution. So these can result in reduction in osmotic pressure. So when this happens, the water may be squeezed out from the gel, resulting in what we call syneresis, or the whipping effect. So it just comes as a result of the imbalance now between the swelling force and the contracting force of the polymers, due to changes in the osmotic pressure. Now we're saying that, okay, so now we're saying that, how can we therefore reduce or decrease the probability of syneresis? So this is not something that is desirable to happen in a gel. So we're saying, how can we make sure that this does not happen? So we're saying that by addition of electrolytes, addition of glucose or sucrose, or increasing the polymer concentration, these factors reduce the probability of syneresis occurring. And then I think just the last four properties, we're saying, when a polymer is in solution form, when a polymer is in solution, they have a tendency to form complexes in solution. So polymers in solution, they have a tendency to form complexes. So these complexes, sometimes they can be used for delayed drug release, because they are not easily dissolved, or they are not easily destroyed in the body. So we're saying polymers in solution, they have an ability or a tendency to form complexes in solution. For example, when a high molecular weight polymer of polyacids are mixed with polyethylene glycols, which are the PEGs. So when these two polymers are mixed, they have a tendency to form complexes. And then apart from that, polymers in solution, they have a tendency to bind to ions. When a polymer is in solution form and ions are added to them, they have a tendency, to be bound. So for example, and because of this interaction of your polymer solution and let's say ions, they tend to form gels. So another way to formulate gels is just to introduce these polyvalent ions, in a polymer solution, and they're going to formulate gels. Though it's not all of them, there are certain polymers that can react with charged substances or charged molecules to form a gel. So for example, calcium alginate is a gel. So in alginate solution, to this solution, when calcium is introduced in here, it tends to form a gel. And then the other one is that polymers in solution, they tend to adsorb at the interface. So if you remember, during drug stability, we looked at this word adsorption. So we said sometimes like plastic containers, they have they can absorb the drug. Okay, so it means the drug gets sucked into the plastic container or just the packaging. So polymers that are in solution, they have actually tendency to absorb at the interface. So what interface are we talking about? So they tend to absorb to the packaging, okay, whether it's the glass or whether it's the plastic packaging. So we're saying, for example, insulin, to be specific, it absorbs to the inner surfaces, of the glass. If it's in glass, it will just absorb there. It will be like it's going to be sucked into the glass, or it's going to be attached, or it's going to adhere, okay, to the glass or the plastic tubing giving sets. So these are, those things when someone they say no, give them IV solution. So sometimes to that IV solution you can add insulin. So sometimes insulin have a tendency to stick to the giving sets. Now, what is the implication of this? It reduces the concentration of the insulin that your patient receives. And then so we're saying apart from that, we're saying it's not every time that adsorption at the interface is bad for these polymers. Of course, for insulin, it's bad because it reduces the therapeutic effect. But sometimes these polymers can also adsorb at certain. So an interface is just a separation between two phases, like at a boundary between two phases. So, for example, here, gelatin, polymers like gelatin, polymers like acacia, polymers like polyvinyl alcohol, they can also adsorb at the interface of the dispersed suspension particles. So let's say you have a suspension, so you're going to have, let's say, your particles, whether they're drug particles, dispersed in a liquid. Now in this liquid, for these particles to remain dispersed in a solution, okay, you need to add a suspending agents, or for them to remain suspended, you need to add a suspending agents, which is going to reduce the probability of them aggregating. Okay, so resulting in stability of these colloidal dispersions or these dispersed systems. And then the last one, polymers in solution, they have a tendency to imbibe large quantities of water, or they have a tendency to absorb, okay, or to attract large quantities of water, okay, and because of this, because they have an ability to imbibe, larger quantities of water or solvent, these polymers have then been used in the formulation, of towels, okay, of nappies, okay, and of surgical dressings. For example, bandages. That's why they put bandages on oozing wounds, because they are made of polymers that have an ability to, imbibe large quantities of water. And apart from that, these polymers have also been used in the treatment of constipation. So constipation is just it's a condition where you're not able to pass let's say stools, maybe for a week or so, even just maybe 4 days or so. So what happens is orally you're going to take this polymer. Okay, now what is going to happen? So it's not going to be absorbed because it does not dissolve. So when it reaches in your intestines or in your GIT, it's just going to remain there in the intestine. It's not going to get absorbed. But then what is it going to do? So it's going to attract water from the body into the intestines, okay? And then as the water accumulates, because it has an ability to imbibe large quantities of water, so as the water accumulates now in the intestines, okay, it softens the stools, and then they are easily passed out. So that's how it's used in the treatment of constipation. So this brings us to the end of the solution properties of polymers. So in the next lecture, it's just about the polymers that are used in medicine. So the water soluble, and also the non-insoluble water polymer. So for those ones, there's no need for me to make a video because there's nothing that I have to draw. So I'll just send an audio for those ones. But then the last one, which has to do with applications, that one I'll send a video, because I'll have to do demonstrations on how these drug delivery systems that are made of polymers work or deliver the drug. Okay, so that's where we're going to end.